Bismillahirrahmanirrahim. Assalamu alaikum. Today we are going to study about the Mendel's law. Gregor Mendel was the first person who introduced the experimental genetics, and he started in an abbey garden. As you know that heredity is the transmission of the trait from one generation to the next. The field of genetics, the scientific study of the heredity, begins in 1860 when an Augustinian monk named the Gregor Mendel deduced the fundamental principle of genetics by breeding the garden pea. Mendel believed and worked in an abbey garden in Austria, and he is strongly influenced by his study of physics, mathematics, and chemistry at the University of Vienna. His research was both experimentally and mathematically rigorous, and these qualities were largely responsible for his success. In a paper published in 1866, Mendel correctly argued that parents passed on to their offsprings discrete heritable factors, and at that time he didn't use the word gene. At that time he used the word heritable. There are heritable traits or heritable factors that are responsible for the transmission of certain traits. In his paper, Mendel stressed that the heritable factors, today called genes, retain their individuality generation after generation. That is, genes are like the playing cards. A deck may be shuffled, but the card always retain their original identities, and no card is ever blended with another. Similarly, genes may be rearranged, but each gene permanently retains its identity. Mendels probably chose to study the garden pea because they had short generation time, produced large number of offsprings from each mating, and came in many readily distinguishable varieties. For example, one variety has a purple flower and another variety has white flower. A heritable features that varies among the individuals, such as flower color, is called a character. Each variant for a character, such as purple or the white flower, is a trait. Perhaps the most important advantage of the pea plant as an experimental model was the Mendel could strictly control. As you can see in the right side of the figure, there is a pea flower. There is a pea flower representing the different flower parts: stamen, carpel, and petals. And this part of the pea flower almost completely enclosing the reproductive organ, the stamen and carpel. Consequently, pea plant usually are able to self-fertilize in nature. That is, sperm carrying pollen grain released from the stamen land on the egg containing the carpel of the same flower. Mendel could ensure self-fertilization by covering a flower with a small bag so that no pollen from another plant could reach the carpel. If Mendel has to be self-fertilize a plant, he covered the plant with a bag. And when he cross fertilized, what he will do? When he wanted cross fertilization, he used the method shown in the figure 9.2. As you can see that here is a figure in which the method is explained how the Mendel self-fertilized, cross-fertilized the plants. He prevented the self-fertilization by cutting off the immature stamen of the plants before they produced pollen. In first step, you can see that he removed the stamen, immature stamen. In second step, to cross-fertilize the stamen-less flowers, he dusted it carpel with the pollen from another plant. And after fertilization, in the third step, the carpel developed into the pod containing the seeds. And in the fourth step, he planted the seed. The seeds grow into the offspring plants. Through these methods, Mendel could always be sure of the percentage of the new plants. Mendel's success was not only to his experiment approach and choice of organism, but also to the selection of the characteristic to study. He chose to observe the 7 characters, each of which occurred as two distinguishing traits. There is seven different characters which the Mendel uses in his experiment. The first one is flower color, flower position, seed color, seed shape, pod color, and stem length. These are the seven different characters which the Mendel used during his. Now, Mendel performed many experiments in which he tracked the inheritance of character that occur in two forms, such as flower color. The result led him to formulate the several hypotheses about the inheritance. Let's look at some of his experiments and follow the reasoning that led to his hypothesis. In figure 9.3a starts with a cross between a true breeding pea plants with a purple flower and the true breeding pea plants with white flower. This is called monohybrid cross. And why we called it monohybrid cross? Because we are taking only a single trait. When we are taking single trait, this type of cross is called monohybrid cross. Because the parent plant differ in only one character, flower color. Mendel observed that F1 plants all had the purple flowers. They were not light purple as predicted by the blending hypothesis. Was the white flower plant's genetic contribution to the hybrid lost? By mating the F1 plants with each other, Mendel found the answer to be no. Out of 929 F2 plants, 705 had the purple flowers and 224 had the white flowers, a ratio of about 3 plants with purple flowers to every one with white flowers. Mendel reasoned that the heritable factor for the white flowers did not disappear in the F1 plants but was masked when the purple flower factor was present. He also deduced that the F1 plants must have carried two factors for the flower color character, one for the purple and one for white. Mendel observed these same pattern of inheritance for six other P characters. From his result, he developed four hypotheses described here using the modern terminology such as gene instead of heritable factors. Here are the four hypotheses which was deduced by the Mendel after their experiment. And he said there are the alternative version of the gene that accounts for the variation in inheritance character. For example, the gene for the flower color in the pea plant exists in two versions, one for purple and other for white. The alternative version of a gene are called allele. So there are uh, two alternative form of a gene is present, and that alternative form of a gene is known as allele. And the second hypothesis was for each character an organism inherit two alleles, one from each parent. These alleles may have be the same or the different. An organism that has two identical alleles for a gene is said to be homozygous for that gene. An organism that has two different allele for a gene is said to be heterozygous for that gene. He also proposed the idea of heterozygote and homozygote. And third hypothesis was if the two allele of an inherited pair differ, then one determines the organism appearance and is called the dominant allele, and the other has no noticeable effect on the organism appearance and is called the recessive allele. We use uppercase letter to represents the dominant allele and lowercase letter to represents the recessive allele. So after the work, he also deduced or give an idea about the recessive and dominance. A sperm or egg carries only one allele for each inherited character because allele pairs separates from each other during the production of gametes. This statement is called the law of segregation. When sperm and egg unite at fertilization, each contributes its allele, restoring the paired condition in the offspring. And you can see in the figure 9.3b explain the results of in figure 9.3a. In his example, the letter P represents the dominant allele. Capital P represents the dominant allele and the small p stands for the recessive allele. Both parental plants were true breeding, and Mendel's first two hypotheses proposed that one parental variety have two alleles for purple flower and other had two alleles for white flower. Consistent with hypothesis 4, the gametes of the Mendel parental plants each carry one allele. Thus, the parental plant in figure 9.3b are either small p or the capital P. As a result of fertilization, the F1 hybrid each inherited one allele for the purple flower and one for white. Hypothesis 3 explains why all of the F1 hybrid had purple flowers. The dominant P allele has its full effect on the heterozygotes, while the recessive P allele have no effect. Mendel hypothesis also explain the 3:1 ratio in F1 F2 generation because the F1 hybrid are small p capital P. They make the gametes capital P small p in equal numbers. The bottom diagram in figure 9.3b called the Punnett square shows the four possible combination of the alleles that could occur when these gametes combine. The Punnett square show the proportion of the F2 plants predicted by the Mendel hypothesis if the sperm carrying allele P fertilize an egg carrying the allele capital P, the offspring will be the capital P will produce the purple flower. Mendel hypothesis predict that this combination will occur in 1/4 of the offspring as shown in the Punnett square. The hypothesis also predict that 1/2 of the offspring will inherit one P allele and one capital P allele and one small p allele. These offspring will all have the purple flower because capital P is dominant. The remaining 1/4 of the F2 plants will inherit two small p and will have white flowers. Because an organism appearance does not always reveal its genetic composition, geneticists distinguish between an organism physical traits called the phenotype and its genetic makeup, its genotype. So we see that figure 9.3a shows just phenotype, while figure 9.3b shows both phenotype and genotypes in all sample cross. For the F2 plants, the ratio of the plants with purple flowers to those with the white flowers 3:1 is called the phenotypic ratio. The genotypic ratio as shown by the Punnett square is 1 is capital P 2 capital P small p and 1 small p. Mendel found that each of the 7 characteristics he studied exhibiting the same inheritance pattern. One parental trait disappeared in F1 generation only to appear in 1/4 of the F2 offsprings. The mechanism underlying his inheritance pattern is stayed by the Mendel law of segregation. Pair of alleles segregates during the gametes formation. The fusion of the gametes at fertilization creates the allele pair on once again. And research since Mendel experiment has established that due to the separation of the homozygous chromosomes during meiosis II, the law of segregation applies to all sexually reproducing organisms including humans. And that's all. Thanks for watching.